Okay, people, shall we get started? Is everyone here? All right, welcome to the last session of this conference. So let's try to be awake and alive for this session. We have some interesting talks. Our first speaker is Thierry. I am not sure if I want to hazard pronouncing his last name, but I will try. Bakel Jiao. I hope I got it right. Thierry is a professor at the Royal Belgian Institute of Natural Sciences at Brussels. And his research focuses on the taxonomy, phylogeny, and population genetics of animals, in particular molluscs. His talk title is What's in a Name? Reflections on defining and naming genera using molluscs as an example. That should be an interesting talk. Thierry, please take over.
Thank you very much. I'm going to put first the screen in full modus. So, first of all, I would like to thank Aravind and the Congress Committee for inviting me to participate in this Congress. I really felt honored that you asked me, but at the same time, I'm really regretting very much that I can't be there in person for the simple reason that, just like Tom, I didn't manage to get a visa, and I'm still waiting for the second application. And I think, well, it will arrive a little bit too late anyway. I also would like to thank you very much for honoring me with the award. And again, it's a pity that I could not be there to receive it. I'm not sure whether I deserve it in the shadow of people like Henry Godwin Austen and so many other illustrious malacologists who have been working on the malacofauna of Southeast Asia or South Asia. But I'm feeling really honored and flattered, and so thank you very much. And it gives me another reason to come and visit you, if it was not just to pick it up, basically. So here I will, my idea of the talk here is to discuss a little bit and to reflect a little bit on how we, on the practices of naming a genera. Of course, I will only briefly touch things. And I do that here with my co-authors and friends. I must immediately say that not necessarily all the views that I will present here are necessarily endorsed by them, but it's by discussing with them and by the exchanging with them and with many others that I gradually try to come to these reflections. And here, yeah, well, with this emoji, I want to express two things. Now, firstly, also the fact that I can't be there, but secondly, also the fact that I suppose I will not tell anything new to you on one thing. And on the other hand, I probably will not resolve anything. Instead, the idea of my talk is to draw attention to some current practices with respect to genus level taxonomy, current practices that actually are a little bit erratic, are a little bit problematic. And so the aim is not, I will discuss or I will present a number of examples of cases where I think there is a problem. And it is not the idea to blame people or to offend people or to upset people. No, certainly not. The idea is really to come to a friendly discussion and friendly reflections on some issues that I think are important for taxonomy and for the users of taxonomy. And basically, I just emphasize here, even if people think that I am offending them, so well, let me be very clear. Actually, I'm blaming myself. I'm in many cases I'm guilty as charged as well. What is also important to know is that what I will present is a work in progress. It's still going on. And I hope that in the near future, I may produce some little paper or some opinion paper about things, but that will, well, we'll see. And so I stress the views I present are not necessarily shared in all aspects by my friends and co-authors that I just showed. Now, what is the issue? Well, actually, I would like to start by the idea, very basically, that when we describe biodiversity, we do that in terms of species. And species they are characterized by scientific names, and these scientific names rely on the initial work, on the initial invention, basically, of Linnaeus, the binomial nomenclature. And in the binomial nomenclature, a scientific name consists of two parts. You have the first part, which is the genus name, and that is then followed by a specific epithet specifying which species is involved. This means actually that when we describe species, the description of genera and the introduction of generic names is an important issue, is an important aspect of naming species, because the genus name provides the first element in a species name. So it is an important act. Now, the interesting thing is that when we look at species, then basically we are dealing with a category, a biological category, an entity, whatever you like to call it, for which there are, I think at this moment, there are more than 37 different species concepts. That means different ways as to how you can define or delimit species. What are the characteristics of this category? Or what are the kind of arguments upon which a species is recognized? So there is a quite strong, quite extensive theoretical framework to define species or to interpret species and to characterize them. In contrast, for the genus level of category, when that's not only at the genus level, also for more inclusive taxa, such a conceptual framework is largely lacking. There is no. If you ask people what is a genus, it would be very difficult to provide a generally accepted definition or even a kind of a clear conceptual framework. Sometimes people have tried to define genera on the basis of, for example, hybridization. In that sense, they would think that species within the same genus are able to interspecifically hybridize, while species from different genera would not be able to do that. But if you think about the difficulties with respect to interspecific hybridization at the species level, you can say that actually the idea of this hybridization criterion is not really very convincing to define genera. So a more general definition that sometimes is used, or some more general concept that is used for the genus is written here. A genus would then be defined as a group of species that are more closely related to one another than they are to any species assigned to another genus. And that circumscription, that kind of delimitation or that kind of description of a genus is then associated with monophyly. Genera should be, in principle, monophyletic. Now, here, the problem is actually that if you look at this definition, we see that this kind of framework, or this kind of concept of a genus actually is applicable to nearly any other more inclusive category. You could use exactly the same definition for example, a family. You just replace the word genus by family, and actually it would work as well. So it's not a very specific concept that aims at the genus level. Actually, it is a more general concept that can be applied to any more inclusive category. But what is important is this link between the genus idea and phylogeny. So currently, in current practice in taxonomy, it's very important to see that there is a link between genera and defining genera and phylogeny. And it's very important also to keep in mind that in the past, let's say up to halfway the 20th century, this was absolutely not the case. And so it means that a majority or a large number of genera have been defined, have been delineated without phylogenetic considerations. And that brings a number of problems. And one of the reasons that people wanted to define or want to create new genera is indeed the fact that the genus name is important for the species name. It's the first part of the species name. And why will people, or why do we try to differentiate between different genera? Well, there are various reasons or various rationales that can be proposed. First of all, for example, people say, we have to create new genera for species that are unique, or a group of species or species that are unique, that are very different from the others. And that often leads them to the creation also of monotypic genera. Maybe we come to that point a little bit later. Another motivation for creating a new genus or for describing genus is the degree is because the species are very divergent. They show a large divergence towards other species, or at least supposedly. So that can be a ground to create monotypic genera. A third motivation could be, the species I find here a new species. Well, it doesn't fit into any existing genus. I don't see any match with an existing genus, so I have to create a new genus. Also, that quite often leads to the creation of monotypic genera. And a fourth possibility is, simply, very pragmatically, my species needs a name. And given that I can't assign it to anything else, I create a new genus. Also a reason to create monotypic genera. Evidently, these four reasons, these four motivations are interlinked and are not independent. Uniqueness is, of course, also probably reflecting a degree of divergence or whatsoever. Or if you cannot fit a certain species into an existing genus, then it is probably because it is very different or very unique or whatsoever. Two other rationales are, for example, an existing genus is considered to be too large. There are too many species in a genus, so authors or researchers might say, okay, this genus is too large, we have to split it into a number of smaller genera. It's a practice that is applied, and I will give an example of that. And a next motivation could be, we create a new genus just to reflect scientific progress, or to reflect new insights we obtain. And we transform these new insights, or we are going to advertise these new insights by creating a new genus, or that can be a higher or more inclusive taxon. So yes, while you could understand from a pragmatic point of view these motivations, these rationales to define genera, but at the same time, it's very clear that these motivations, these reasons, are very arbitrary and definitely highly subjective. And because of these characteristics, this practice has implications for three important issues. Firstly, the epistemology of taxonomy. What is the kind of knowledge we obtain by taxonomy, and how do we obtain this knowledge? And we see here that there is a strong subjective element in it, and that, of course, is from scientific reasoning, from scientific point of view, problematic. Another implication is for the users of taxonomy, that are all those people who are not taxonomists, but who need to work with the taxonomy, who need to use the scientific names of taxa, or need to know the scientific name of species. And they, of course, they will depend strongly, or the implications of this practice are indeed affecting the users and the users community, or so-called, in a nice term, in management terms, the stakeholders of taxonomy. And a third element for which these practices can have severe implications is for the credibility of taxonomy itself. And that depends, of course, largely because of this element of arbitrariness and subjectivity. And it was already outlined by Bernard Palgergeli earlier this week. Indeed, in taxonomy, there is a strong element of subjectivity or personal opinion, a matter of opinion. And that indeed is sometimes by non-taxonomists perceived as a flaw in taxonomy. And it could lead to a kind of a lesser appreciation for taxonomy as a fundamental science. And what I think is important to stress here, during my talk or during this reflection, I consider taxonomy as an extremely fundamental science, extremely important for several reasons. But one of the reasons is also it is actually a heuristic tool to keep track of biodiversity. And I stress the element of heuristic. And heuristic is a mental shortcut used to simplify problems and avoid cognitive overload. Indeed, the number of names we can manage and so on is limited. So an important issue for taxonomy is to not overload all the users of taxonomy with a plethora of names that can no longer be managed, and that actually do make it very difficult to manage and to deal with that extreme biodiversity that we know. So I think it is a heuristic tool. It's very important. And so I would like to discuss now and reflect a little bit about these issues using these features by presenting three issues, and they are interconnected, three interconnected issues. And the first of these issues actually is the splitting of genera. And as an example, I take here the split of the oyster genus Crassostrea. This split, the splitting up of this genus was proposed in 2017 by a group of Italian researchers who really performed an excellent, marvelous study, molecular study of the genus Crassostrea. And on the basis of that study, they proposed to split it up in at least two major clades. And yes, the genus Crassostrea, you probably all know it. It is the genus in which for example, to which the Pacific oyster, Crassostrea gigas, has been assigned. And the type species is Crassostrea virginica from the Atlantic Ocean. And what was the general idea? Well, before 2017, the genus Crassostrea was known, a very well established genus, including Crassostrea gigas and Crassostrea virginica. And that was done, it was a practice that was already taking place for more than 50 years. It was very stable. And yeah, Crassostrea gigas, being really one of the two most important aquacultural species, was placed in this genus. And what was the general idea of the Italian group, of the Italian show, was actually the Crassostrea, the genus Crassostrea, as it was known up to then, should be split into two major clades, a Pacific clade and an Atlantic clade. And what was the result? The Atlantic clade, that's also the clade in which Crassostrea virginica is placed. And Crassostrea virginica is a type species of the genus. So what they did was restrict the genus name Crassostrea just to the Atlantic clade, and then create a new name for the Pacific clade, and that would then become Magellana. With as a result that this very famous Crassostrea gigas had to be renamed to Magellana gigas. I stress here, it was based on really very nice molecular data. Just to be correct, this Pacific clade actually is not only the genus Magellana, but is also a very small genus Stalanostrea that was described on the basis of some morphological characteristics, not very clear, but we leave that a little bit out of the discussion for the time being. Now, so as I've mentioned, Crassostrea gigas is a very well-known species. It's one of the most important aquacultural shellfish, together with the Manila clam, and there is some competition between the two, depending on what characteristic you're looking at, in terms of tonnage or the financial amounts that are involved. And here you see the distribution. The native range of Crassostrea gigas is here in the waters around Japan, but it has been introduced elsewhere in the world where it is really used, is used for aquaculture and for production, for consumption, for human consumption. And in fact, this map is still not giving a complete picture because, for example, Crassostrea gigas is also here common along the southern coast of the Mediterranean, where it's also used for aquaculture purposes, is also along the coast of the Levant, and it's also very common and widespread in the Black Sea, where also now attempts are being done to use it for aquaculture purposes. So you could actually say Crassostrea gigas is a kind of, if you like, some sort of a Drosophila in the bivalves. The amount of literature about Crassostrea gigas is huge, is absolutely huge. And really the name Crassostrea was well established. And so the splitting up of the genus Crassostrea, such that Crassostrea gigas had to be renamed Magellana gigas, provoked a major upheaval in the community of the workers on oysters. And here you see the data, or some of the data on which it was based. You have here in blue the Pacific clade in Crassostrea. So this is the complete genus Crassostrea, the red and blue together. That's the former genus Crassostrea. And that was split on the basis of these data here mitochondrial DNA data, here nuclear DNA data, and it was split consistently and highly supported into two clades, namely the Pacific clade Magellana, that had to be renamed Magellana, and then the remaining Atlantic clade that kept the name Crassostrea. That picture here is also mirrored here in the nuclear tree. Again, here the clade, the Pacific clade Magellana, with here the Atlantic clade Crassostrea. Very important. Look at this clade here. In total, the overarching clade, and this was the overarching clade that until then was named Crassostrea. Further data were added to it. Here, for example, complete mitochondrial genomes, and there it was noted that between the Magellana clade and the Crassostrea clade, there were a number of gene order changes occurring in the mitogenomes, also highly supported, and so very nice data, basically. And yeah, from the moment that the Italian group was publishing their ideas about the phylogeny of the genus Crassostrea, the logical consequence was that Crassostrea gigas had to be renamed to Magellana gigas. And yes, this provoked this upheaval in the community of the people working on oysters, and people were really upset, and it became really a very acrimonious discussion. People were complaining, and they didn't like the idea, and they say, why changing this well-established name, and so on. And so the question arose, yes, what was the need, or was there a need to create this new genus? Was there a need to split up Crassostrea into different genera? What is the added value of creating this new genus? And there, a first point that is important to see is that this divide between the Atlantic clade and the Pacific clade actually was already known for quite a long time. Already in 1993, it was observed that there was a kind of a hybridization barrier between the Pacific oysters and Atlantic clade, between the Pacific clade and the Atlantic clade. But there appeared to be no or very little hybridization barriers within each clade. So interspecific hybridization was possible between species within the Pacific clade, and interspecific hybridization was possible between species within the Atlantic clade, but there was no interspecific hybridization possible between species from the Pacific clade and the Atlantic clade. So that was already observed quite early in the 90s. Next, 2 years later, it was published that there was a phylogenetic analysis of the mitochondrial 16S RNA published, and that analysis showed very nicely, indeed, that there was a divergence between two clades, namely the Pacific clade and the Atlantic clade. But that was known. And in early years 2000, there was another study showing that at the karyological level, at the chromosomal level, using fluorescence in situ hybridization, it was showed that there were very consistent, clear karyological chromosomal differences between species in the Pacific clade and species in the Atlantic clade. Now, what is important, these studies indeed clearly indicate already in those days that in the genus Crassostrea, as it was known, was divided into two subclades. And there were two groups, a Pacific group and the Atlantic group. But none of these studies, none of these authors expressed the need or proposed the idea to split Crassostrea to reflect these facts. It was not perceived necessary to do so. And now if we see at how the reaction was of worms, the World Register of Marine Species, and mollusca base linked to it, specifically about the molluscs. Well, their first reaction was, it's their policy, in 2017, when the split was proposed, they took over that opinion. And so when you look at that moment into worms, and you wrote, you were looking for Crassostrea gigas, you would get this message, no matching results found. So immediately the split was implemented and taken up by worms. And then because of the major upset provoked in the oyster researcher community, then worms adapted this position and changed this feedback into when you typed Crassostrea gigas, then you would receive this message: alternate representation, slightly less preferred. That is an addition which I always find a little bit difficult because it's never clear why it would be slightly less preferred. But anyway, so an alternate representation. But then in 2021, the Italian researchers published a rebuttal of the complaints of the oyster researcher community. And in that rebuttal, they provided new evidence, nice evidence, for example, the mitogenomic evidence. And after that paper, worms changed its attitude again, and it was now very definite. If you typed Crassostrea gigas, and if you do it now, then you will get the feedback unaccepted. So the question now is, okay, what is now in this practice the elements? What are the elements pro and contra splitting up this overarching genus Crassostrea into two genera, Crassostrea and Magellana? Let's look first at the arguments pro or supposedly to be in favor of the split and the recognition of Magellana. Remember, we have overarching genus Crassostrea divided into clades. One clade would be then Crassostrea sensu stricto, the other clade would be Magellana. So what was the first thing that the Italian researchers claimed in their rebuttal? They said, well, but this name change is not a problem. It's not a big problem for users, which is a little bit a strange argument because it were exactly the users who were complaining. So apparently the users perceived it as a problem. But anyway, so they said, no, the name change is not a big problem because in indexing services like worms and mollusca base, it is easily implemented. You can easily change it, and so no problem. Now, this is indeed true, but this is not true for all indexing services. And evidently, if worms and mollusca base can act and can adapt themselves immediately to this name change, it's quite logical because worms and mollusca base are specific taxonomic databases. These are databases whose own purpose is to provide a taxonomic backbone. So it is quite logical that they are able to implement this name change immediately. But other very important databases, like GenBank, do not do so. And hence, it means that the name change proposed by the Italians, so to split up the former overarching genus Crassostrea into a genus Crassostrea sensu stricto, Atlantic clade, and the genus Magellana for the Pacific clade, is not implemented in GenBank. It's only taken over depending on how the authors submit their data. So if you want to download all the sequences of the species gigas, you have to know that, okay, I have to download all the Crassostrea gigas and I have to download all the Magellana gigas to be sure. So it's an element pro if you look from the position of worms and mollusca base, but it is a little bit less convincing when you look at several other important databases or indexing services. A second argument that was taken as a positive pro-argument was, well, the tree clades are phenotypically diagnosable. And here I have to say, yes, they are phenotypically diagnosable. First question you could ask is, okay, but is that the reason to create new generic names for them? That's one thing. Secondly, the diagnosability that we're referring here to is a molecular diagnosability. It is not a phenotypic, at least not a morphological diagnosability. You can discuss about, I have nothing against molecular diagnosis, on the contrary, but that's another issue about which one can discuss. But in this case, actually, the phenotypic diagnosability refers to molecular characters. For example, the gene order changes in the mitochondrial genomes. A next argument in favor of the Crassostrea thing was to realize that there was a deep divergence between the two genera. So the split between the genus Crassostrea sensu stricto and the genus Magellana, so the Pacific and the Atlantic clade, that divergence is deep. Okay, that is indeed a correct observation, But again, one should ask, is that a good argument to say, so we have to give new generic names? Then a fourth argument in favor of Magellana is, well, recognizing the three genera would be a more stable taxonomic choice. Here, my question is it? And I look for good reason to say, why would three names be more stable than using one name? I'm not convinced about that. But yes, okay. And then lastly, it is said, yes, taxonomic names, species names, are hypotheses. They are hypotheses. And my question here is, yes, is that indeed the case? And if in the case of Magellana and Crassostrea, what is the hypothesis and how do you test and reject it? I'll come back on this in a few moments. So what are now the arguments against the idea of creating Magellana and keeping the overall genus Crassostrea as it was in former times? So keeping the two clades together, the Pacific and Atlantic clade together in one single overarching genus Crassostrea. Well, the first argument in favor of that is basically the excellent work produced by the Italian group, really very nice data, is absolutely marvelous, but in fact only confirmed what was already known before that time. They confirmed the fact that indeed there are two clades and that these clades are well supported. That's a nice confirmation, but basically, from the phylogenetic point of view, nothing was changed by the results of the Italian research. So nothing changed. So that means, in fact, if you, and I'm going in the back, if you look back at the trees here, look at the overarching genus, well, the genus, the joining, the sister group relation between the Pacific clade, Magellana, and the Atlantic clade, Crassostrea, is maximally supported. And that was it before. And also in the nucleic genes, you see, it uses an extreme high support. So basically, the overarching genus Crassostrea was not changed. That clade, that overarching, the larger clade, was still there. So there was no new phylogenetic insight. It was only a confirmation of what was already known quite before. So yeah, basically the overarching genus Crassostrea sensulato basically was not changed, there was no problem with it, it did not become paraphyletic or no, nothing. It remained exactly a monophyletic group, a clade, with two subclades. And I'm sure when you look further into the subclades, you will find subclades, no problem. So that is a page where why would you disturb a very stable taxonomic assignment, and why would you split it up, making life more difficult for users? The second reason why I would indeed advocate to keep Crassostrea is, well, there is a problem with this hypothesis thinking. Does the Magellana hypothesis, does that hypothesis refute the Crassostrea sensulato hypothesis? I don't think so. Nothing has changed. So I would not see how this idea that it is a hypothesis would make Magellana a more or a better hypothesis or than the Crassostrea overarching. And the third very pragmatic reason why keeping Crassostrea as it was is when you look at the information content and the didactic value of the names used. And there, for me, the question is, what is the most interesting, the most relevant for the users of taxonomy is using a new name, Magellana, or Talanos, but the Talanos will leave it outside, using the name Magellana, which doesn't tell you anything, basically, or saying, oh, we have the genus Crassostrea, and within Crassostrea, there is a Pacific clade, and you use the name Pacific clade. I think for users of taxonomy, that's a more logical and much more informative approach than introducing a new scientific genus level name. Secondly, by splitting up the former Crassostrea sensulata into a Crassostrea sensu stricto and a Magellana, you create confusion because you have now two interpretations of Crassostrea, which will actually not necessarily be distinguished, particularly not in the literature. So you have a Crassostrea sensulato referring to the overarching clade, including the two subclades, the clades Pacific and Atlantic, and the Crassostrea sensu stricto, which then only refers to the Atlantic clade. So for the users again, that makes life more complex, more difficult. And a third, okay, it is not very big mathematics, but okay, adding new names increases, of course, taxonomic complexity, and particularly for the users again. One name, Crassostrea, versus 2 or 3 names, Crassostrea, Talonostrea, and Magellana. So what's the most easy for the users? If nothing from the phylogy point of view has changed, you can discuss about that. And then the last element to maintain Crassostrea is a simple, very pragmatic, again, user's point of view, is that changing scientific names in trade or in legislation or in indexing is always a little bit problematic. I already referred to the problem in GenBank, but also in trade and in legislation, new scientific names will find much slower their way and their general acceptance. And that can create indeed problems from a purely pragmatic point of view for the users. So what's the conclusion here? Well, actually, there is no clear conclusion. In fact, Worms decided Crassostrea gigas is an unaccepted name, should now become Magellana gigas. But within the reflection I gave here, one sees that the discussion stays. And in fact, a large part of the oyster researcher community simply do not want to use Magellana gigas and keep on using Crassostrea gigas. For me, this means that actually we, as a taxonomist, we should reflect a little bit on what are the consequences of the actions we propose, or the consequence of it. And in this case, one could easily indeed question what is the added value, what was the need, what was the necessity to split up a well-known genus into 2 genera and to introduce this new genus name Magellana for a species that is so commonly used and so commonly so important in the world trade. You can wonder, is it relevant to really to introduce this kind of new names? So okay, this brings us actually to the second part, the second issue I wanted to discuss. Oh no, first another example of what is the result of splitting genera. Here, just as an example to what it can lead. You probably all are more or less acquainted with the family Conidae. It's a marine family of poisonous snails, carnivorous snails, hugely diverse family, at least in terms of species. There are about between 800 and 900 species known. And in former times, all these species were referred to one single genus, the genus Conus. And in fact, I think about 95% of all the species, when you get them, when you see them, you would immediately recognize that's a Conus. That was easy in those days. There are a few species that look a little bit more aberrant, but that's not really here. That's a little bit, but anyway. But so from over the years 2000, people started working, and it was perceived that the genus Conus, with more than 800 species, was too large, was not good. So at a given moment, a group of researchers split up the genus Conus into 115 different genera. And you see here the phylogy tree of a series of these genera. Several of these genera were monotypic, by the way.
Can I just interrupt?
The question that we see here is, or that you can ask here is, what is the added value of this split up? For all the users of Conidae, it was so easy, at least for me as a non-specialist.
Can you hear me?
I could immediately, this is a Conus. The genus Conus, with its 800 or more than 800 species, was a monophyletic entity. No problem, no discussion about that. It was a nicely well-defined monophyletic group. It was a clade. But so now with this 115 genera, you got a far more complex assemblage of names, of course. All these genera, and it became extremely difficult with all these generic labels. And so in 2014, a group of researchers from from the Paris Museum reassessed this phylogeny, and they reduced then again the 115 genera that were proposed. They reduced them to 4 to 6 genera, which at least is a little bit showing far more common sense, and I think is still a far more practical solution to deal with the taxonomy of conus than these 115 genera. But the all-over question remains, why was there the need to split up the genus and to create new genera for something that was actually very stable? It's a nice, an extreme example, but an example. And it still goes on. People might find that a genera may be too large or too, and then be seduced to split up that these genes. Okay. Now, this brings us to the next issue that I want to deal with is actually, yes, it was used by these Italian researchers looking at Crassostrea, and they found, okay, there is a deep divergence. These two clades, the Pacific clade and the Atlantic clade, they are deeply divergent. So we have to create new genera for that. And in general, we quite often will first recognize taxa because they are somehow different from each other. And that's a logical and a very normal practice. But the perceived, I have to, yeah. Okay, perfecto. Okay. Perfecto. I'm near the end. Okay. So I will not go too far. The two other issues I wanted to deal with here is the fact that, first of all, we use big differences as a kind of as a means to define genera or other higher or more inclusive taxa. What I want to say is that the big problem with this practice is that if genera are reflecting phylogenetic relationship or should be based on phylogenetic, then actually it's not a big difference, but you should look for synapomorphism. And you should define genera on the basis of common shared synapomorphism between species, not differences and not their uniqueness. And basically, the very important thing to keep in mind is that big differences, and that can I illustrate with, okay, I'm very sorry, these are not mollusks, very big differences. Keep in mind that big differences are not necessarily reflecting deep genetic differences. I take them as a metaphor, the example of a dragonfly. If you look at the dragonfly larvae and you compare it with the phenotype of the adult dragonfly, then you would say this is a huge phenotypic difference. And they live already. This one live in the water. This fly, the adults fly. So huge phenotypic differences, but genetically they represent zero change. Keep in mind that the big differences do not absolutely reflect deep or genetic differences. And the last thing I wanted to present here is the fact that also on the basis of I must see here on the basis of big differences or the uniqueness or distinctivenesses or I find no place for a species, that kind of argument is quite often used to create monotypic genera or monospecific genera that include only one single species. And the problem with this is that such a species, such a genera are not based on synapomorphism. And in fact, the big problem there is that such genera, the species level hypothesis and the genus level are identical. You can no longer actually distinguish between genus and species. And there is a logical flaw. So basically, monotypic genera have no phylogenetic ground, have no phylogenetic meaning, are meaningless. And in fact, are based on the fact that you don't know how to place, where to place it, where to assign it. And so you create a new name, but that name has no phylogenetic meaning, strictly speaking. It has no epistemic meaning, and therefore it's based upon ignorance. We create most of the time monotypic genera because we don't know. And the problem here is, and then I will finish, the problem here is that nevertheless, in other studies, other users of taxonomy will use monotypic genera to take or to study or to infer or to measure, for example, phylogenetic distinctiveness. Well, actually that's not being possible. Or while it is considered that the loss of a species of a monotypic genera is more important than the loss of a species in other genera. And actually it's based simply on ignorance. And for example, in the United States, the Endangered Species Act, there is the monotypic genera or the most genetic distinct group with the highest priority. and they represent unique gene pools of special scientific end. And that actually is not inherent to the idea of the monotypic genus because basically it has epistemic no value. So a very big problem because monotypic genera are highly pervasive in taxonomy. So I'm going to stop here and just to add here an example of an extreme case of a monotypic genus that is problematic is here the Marsupilami. Basically, it is a species that was officially described and accepted. It is published also, it is also covered in the zoological record. But actually, it is the hero of a comic strip album series in Belgium. Les Aventures du Marsupilami is completely invented, but it was completely incorrectly, it was not correctly published as a new species in a new genus with everything that and covered by that case of. And so now I must see that I hope I used the correct language. And then Yavat, thank you very much.
Thierry, can you hear us? Can you hear us? I think he's still muted. Can you hear us?
Yes, I can hear you. Yeah.
Okay. Okay. So thank you for that wonderful talk. There is it's almost a fashion to try and erect new genera because it's a lot more sexy than new species, right? You're going further up. And it's become a disease of sorts. But anyway, let's open this to discussion. Any questions, people? Yeah, there's one there.
Yeah, nice to hear theory. I'm Prem.
Hi, Prem.
I just want to tell you we are missing you here, and we have your gift with us. Only this, not a question, but just to ask you to hello from Bangalore.
Thank you.
Any questions? So while people are still thinking about it, I had a question for you. The oyster example, there was that third genus, I think starts with the T.
The Talonostrea, yes.
What was it? Was that genus already described earlier, or was described as a part of this study?
No, it was described earlier, and it was a little bit, and it was a long time considered. It was not very clear whether it would be taken as a good thing. Yes.
Okay, so in that case, once they had the phylogenetic information, then this particular genus, not the monotypic genus, the Pacific versus Atlantic, that genus became paraphyletic.
Well, no, because in fact, within the Pacific clade, there you have the two clades, the clade Talonostrea, which is a sister group to the Magellana group.
Yeah, what I meant was before Magellana was described.
Yeah.
I forget the older genus. The older name then becomes paraphyletic because this particular genus is nested within. Maybe that was the inspiration for them to split it further.
That could be. It could be one of the reasons to split it. But in fact, the genus Talonostrea is always considered as a little bit not generally accepted. It was a kind of, you say, yeah, okay. So it was not really taken as a strong argument to consider Crassostrea as being paraphyletic. It was considered, these people described it. It's actually described also in Chinese. That was very difficult to access it. So it was a little bit not taken. And I think the motivation of the Italian researchers, and I stress again, their work is really very beautiful. The data are very beautiful. Was actually, I think, they wanted to focus on the new insights. I think that was the major drive, basically. The idea of showing the progress of science.
Yes, that could have been done with just calling it Atlantic clade and Pacific clade.
Absolutely.
So I just wanted to add that we had a similar kind of situation where there was a very distinct monotypic genus, and we built the phylogeny of these lizards called Calotes. This very distinct genus actually fell within Calotes, right? And we just subsumed it into Calotes. and we didn't split Calotus into multiple genera, and we called it subgenus. So we just said, it's Calotus subgenus Brachysaura, because Brachysaura does look very different from Calotus, but it is nested within Calotus. So I completely agree with you. I think instead of splitting and complicating taxonomy more and more, we should keep it simple. I mean, use the more parsimonious approach, as you rightly pointed out.
I think the most parsimonious and also a little bit more conservative, and only go change things when the data are sufficiently strong to support. And there are a lot. So, for example, for monotypic genera, what I would always advise to do is to find the closest genus with which it shares a number of synapomorphies and put it there, even if that's an hypothesis. Okay, no problem. But at least at the moment, and you only split it off when you have far more better data to decide about this.
Yeah, but the Italian group might argue that those mitochondrial rearrangements in the mitochondrial gene order on the lineage leading to Megalana are synapomorphies for that genus.
Yes, I mean that's why they wanted to claim that, and that are synapomorphies. I mean, but every clade is of course defined by synapomorphies. That's the basic idea, and so in this case, I would not deny that there are two clades, and that's absolutely not. That is very, very, very clearly. But the question is, do we need to reflect that by creating new genus names? That's the point. And indeed, as you mentioned, also here, some people have suggested maybe we should simply use the genus name Crassostrea and recognize two subgenera. And that is perfectly possible, absolutely. But then you're sure you create a genus group level name, and taxonomically it becomes equivalent, and sooner or later people might indeed use the two things interchangeably, and then you come again into some confusing situation.
Yeah, I have one more question for you, but I'll let's see if the audience has any other questions. Any questions from the audience? There's one there.
Hi, Thierry. I'm Biju here. My question is that many of the people who are doing integrative taxonomy now argue that the creation of a genus, there is no thumb rule and it should be taxon specific. For example, even within the sharks for recently, creation of normally people say that any genetic difference beyond 3% can be or 10% can be a new genus, etc., etc. And then in that case, will this kind of an argument further create complications in erecting new genus, or can we have an universal, the thumb rule for creating a new genus?
Yeah, I would not. Again, this is a long-standing discussion whether the degree of sequence divergence, whether you can use that to delimit it or to have a kind of an objective tool to delimit certain degrees of taxonomic differentiation. That's the main thing. I would not do that, and because you see that the amounts of sequence diversion are very variable between groups and within groups. And in fact, already, I must confess, when I started doing work, that was a little bit before sequencing time, we worked with Alzheimer data. And in the Alzheimer times, we worked with so-called niche genetic distances. And again, in those days, it was already recognized that actually you cannot use the mere value of niche genetic distance as a measure to say, okay, these are different species or different genera. And in the same thing, in the DNA barcoding community, it's the same thing. It has a long time been tried to find this kind of so-called DNA coding levels that should say that's a different species, but also there it is very difficult, if not impossible, to have a kind of a very objective and consequent scale of amounts of sequence diversion pointing to a certain taxonomic differentiation. I think we should never forget that taxonomy is a human endeavor, and what we try to do with taxonomy is impose boundaries upon entities that actually are constantly evolving. And it is very difficult to capture, and we need these boundaries for our cognitive purposes to be able to handle things, but it's not the way how evolution works. And so I think it is a tricky thing to say, well, we are going to use sequence diversions or this kind of or a measure of difference as a kind of an objective criterion that would be that you can implement throughout all organisms. I think it is philosophically even not possible.
Okay, any other questions? Yeah, so I just quickly, I think, getting back to what you just mentioned, I think we should not even attempt it at coming up with some kind of a genus concept. I mean, beyond species concept, higher levels, it does not make any sense. But what is your view about, so you're okay with naming clades, right? And that's what the phylogenetic nomenclature is talking about, the phylocode. Just getting rid of all the hierarchy. After the species, it's all just each clade is being named based on synapomorphies. So what is your take on that?
Well, to be honest, I am not apparently against the idea of hierarchical structure because that helps us to manage biodiversity. There is a pragmatic cognitive element in having a classification in this way. It helps us to group things, and that is definitely not unimportant. So from that point of view, and I'm saying, yes, I would definitely say, yes, we name clades, but that doesn't mean that all clades have to be named. That's another thing. It's not because we find a clade that we should give a name a priori, because otherwise you come in the same problem and you're inflating the number of names for no added value. I always interested in to say, okay, what does it help us? And that's an important criterion for me.
All right, Thierry, thanks a lot.
Thank you very much. And once again, sorry that I can't be there. It's really a big pity.
Thank you.
Anyway.